in general, autoimmunity will result from some type of failure in tolerance mechanism. So something happens for a failure of self-tolerance. We have self-reactive lymphocytes, and as a result, we see tissue injury and some type of autoimmune disease. That likely involves some type of genetic susceptibility. That also likely involves some type of other environmental insult. These things will come together to alter immune regulation and to help us, sadly, go into autoimmunity. We've already seen some issues related to self-tolerance already in the semester. So when we've talked about tolerance, where have we seen tolerance already this semester? Yeah, Nick? T-cell development. And what kind of tolerance do we call that? Self-tolerance, and there was also another term, central tolerance. So we talked about central tolerance, which is what we see when tolerance mechanisms happen during the development, either during T-cell development or potentially during B-cell development. And so we can have central tolerance mechanisms, and we've already seen those central tolerance mechanisms. So if we recall, this was what we saw with B-cell central tolerance. So when we had a self-reactive B-cell in the bone marrow, it potentially could be deleted. It could undergo receptor editing, or it could be induced to anergy and become anergized. And so we saw this whole process way back when we talked about B-cell development. When we talked about this exact process, we also noted that there was a flaw in central tolerance of B cells. What was the flaw in central tolerance of B cells? Yep.
Not all of the possible cell antigens were present in the bone marrow.
Exactly. So all antigens are not present in the bone marrow. So we only get testing for self-reactivity against antigens that are present in the bone marrow. All other antigens are not tested. And so self-reactive B cells that react to those could, in fact, leave the bone marrow. They would escape a central tolerance mechanism. So all of those B cells that respond to other types of antigens could potentially lead to some sort of autoimmune disease. One additional thing that we need to think about in terms of B cells is the germinal center reaction. So we've talked about the germinal center reaction when we thought about peripheral B cells, where our B cell will be activated and will undergo somatic hypermutation to make variations on the B cell receptor with the goal of binding to antigen with higher affinity. But remember that that process is a process of random mutation. It is possible that in that process, a B cell may mutate so that its B cell receptor now becomes self-reactive. So it wasn't self-reactive when it left the bone marrow, but somatic hypermutation resulted in the production of a self-reactive B cell. And so both of these processes could result in self-reactive B cells being present. We also saw central tolerance, as Nick mentioned, during T-cell development in the thymus. And so if you remember, in the thymus, we also had a little bit of a problem with the scheme. So what was our problem that we saw in T-cell development and T-cell central tolerance? Yep, Nick.
T-cells need to be somewhat self-reactive to the testing?
Yeah, so every T-cell has to have some low affinity for self-antigen in order to be positively selected and to leave the thymus. And so every T-cell is a little bit self-reactive. The ones that are highly self-reactive undergo apoptosis. the ones that are sort of high intermediate become regulatory T cells, but there is some amount of medium binding that allows that T cell to get positively selected. So every T cell is slightly self-reactive. What about the antigen issue that we saw with T cells? So with B cells, we said that every antigen wasn't present. How do we deal with that with T cells? Yep. Yeah. So we have air that leads to the production of the tissue-specific antigens. So we don't have at least one of our problems from B cells is taken care of in T cells, but we still got a problem in T cells. Yep. Air's exact function is unknown. People want it to be a transcription factor, but it will not do transcription factor activities in assays, and so it's not quite clear exactly what it does. So an enzyme kind of works because we don't totally know what it does. So these are all the parts where we've already seen tolerance and when we've already seen central tolerance. There are some situations where patients have defects in one of these processes and thus have what is known as systemic autoimmunity. So if you have a defect in central tolerance, that generally leads to systemic autoimmunity. What that means is that you're having some broad problem with your B cells or your T cells in development, and so you probably don't have one B cell or T cell that slips through the cracks and attacks one organ. You probably have many, and you'll have a multi-organ autoimmune disease where you have many T cells or many B cells acting inappropriately. I told you a little bit about this one disease, APSED, when we talked about this before. This is a condition where patients are missing air, and so those patients are unable to have their tissue-specific antigens be produced in the thymus. So there are whole groups of T cells that don't get negatively selected and get to go to the periphery. So there's no air, it's crossed out. And thus, the T cell is not negatively selected, and it responds to a tissue antigen and leads to autoimmunity. These patients have multiple autoimmune diseases, and each one is a little bit different. So here you can see 85% having hypoparathyroidism, 72% adrenal failure. And so these patients will have sort of a combination of multi-organ autoimmune diseases that will be unique from patient to patient. Most of them will have recurrent yeast infections or candidiasis, and many of them also have nail dysplasia of their fingernails. And so this is one example of what kind of things you can see if central tolerance goes awry. The other really famous type of systemic autoimmunity that we see when there is a defect in central tolerance is called IPEX. IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. And this is a mutation in FOXP3. And so in FOXP3, we have this disease usually seen in boys where they get this list of conditions. So enteropathy, so GI issues, eczema or dermatitis, nail dystrophy again, autoimmune endocrinopathies, so something's wrong with different endocrine glands, autoimmune skin conditions like alopecia and pemphigoid. Pemphigoid is bad. Enlargement of secondary lymphoid organs, insulin-dependent diabetes, food allergies, and infections. Most kids with this die in the first 2 years of life. And so again, here we're seeing multi-organ autoimmunity, multi-organ issues because of this lack of FOXP3 and the inability of these kids to make regulatory T cells. So their T cells aren't going to become regulatory T cells. They might become regular T cells, and they don't have any regulatory T cells to turn off the other T cells. So it's kind of a double-edged problem. Yep. Okay. So we can see situations where central tolerance is sort of totally messed up. But I want you to sit and think for a second about all the ways that B cells can slip through the cracks of central tolerance, or all the ways that you might have an autoreactive B cell in the periphery. all the ways that you might have an autoreactive T cell in the periphery. So let's imagine that we all have good central tolerance mechanisms. Based on sort of these 3 slides, how frequent do you think that autoimmunity might be? Yep.
Probably not super frequent,
Why is that?
because there are ways that the B cells can get energized if they're self-reactive, or T cells can get energized if they're self-reactive.
Well, remember, B cell anergy happens in the bone marrow.
So for B cells, it can happen more than it would happen for T cells, but T
Okay.
cells get energized because they get signal when they're not signal.
Okay.
So if there's no pathogen.
All right, yeah, Vanessa.
If there isn't like an enzyme that's similar to air in the bone marrow, then you might end up with raw reactivity that
Okay.
the B cells just end up.
Okay. Yeah. What were you going to say? No. So oftentimes at this point of the semester, students freak out because they're like, oh my gosh, I'm going to have autoimmune disease tomorrow and I'm going to die. Oftentimes you look at this and you're like, oh my gosh, why don't we all have autoimmunity all the time? We all should have autoimmune B cells and autoimmune T cells based on what we've learned so far that are leading to some type of reaction. And if we look at some of these very common autoimmune diseases, you can see that psoriasis is seen in 1 out of 50 people. Rheumatoid arthritis, 1 out of 100. Graves, 1 out of 100, and so on. The moral of this story is it's not 100% of people. And so from what I've just told you about the flaws in central tolerance, you might look at these data and say, these numbers are lower than I would have expected. I would have expected everybody to have these problems. And the reason why, which was sort of what Mark was alluding to, is because happily, we've got this other thing called peripheral tolerance. So everything we've talked about thus far has had to do with central tolerance. But we do have mechanisms of peripheral tolerance where self-reactive cells can be dealt with in the periphery, as well as in the primary lymphoid organs. So Mark kind of alluded to this a bit already. And in some ways that makes sense. When I first learned how you teach about peripheral tolerance, I learned 6 mechanisms of peripheral tolerance. And I'm gonna write a list on the board of the 6 mechanisms, and we're gonna go through the 6. We'll hopefully make it through most of them today. And if not, that's what Wednesday's for. And what you will see is that the vast majority of them are kind of things you already know. And so in some ways you're going to look at them and be like, oh yeah, I get that. Oh yeah, I get that. Oh yeah, I totally already know that. So like 5 of the 6 you can kind of be like, yeah, I got that. 1 of them you won't, but that's okay. And so some of these are things you've already seen. For each of them, we're going to talk about how we set up that form of peripheral tolerance, as well as how that peripheral tolerance can get broken. So how you could have sort of the bad luck for that peripheral tolerance mechanism to not work, and to have a problem. And so I have these, honestly, the order I have them in is like not the order that I think of them in any, I feel like the order every year I'm like, really, Self? But the reason is because there's actually, the way we break them makes sense in this order. And so, I don't know, sorry brain, it's not the order I like. So if you were going to think about peripheral tolerance, and you were going to guess a way that peripheral tolerance works, based on what you know about central tolerance, what would be the first thing you would guess? Yeah. Okay, so you might guess anergy. Anything else you might guess that's like really simple based on central tolerance? Yeah. Okay, receptor editing. That one doesn't happen. Anergy will happen. Like real simple. Real, so simple. Yeah. Kill it. Deletion. And so one of the things that we see is something called clonal deletion. So deletion can be one of our mechanisms of peripheral tolerance. Sometimes when there is a self-reactive cell, when it encounters antigen, it will lead to apoptosis. The details of this are pretty hard to understand. How does it know if it's a self-antigen versus a non-self antigen? This may be related to some things like chronic exhaustion, of just like how frequently is the antigen seen, how many times is the cell getting hit, because you can imagine a self-reactive cell might be getting hit and seeing its antigen really frequently. And so there's actually a lot that we don't know about deletion. But there's one big thing I want you to think about with regard to deletion. How do you think we break deletion? So let's imagine that you have bad luck and you're going to get an autoimmune disease. Could deletion get broken in you? Could you stop having deletion? How? Yeah. Okay, yeah. If you had a mutation in caspase 3, it would block a whole lot more than just T cells. You would have a whole lot of problems before you got to autoimmunity. Okay? Really, if you were going to break deletion, that means that like a cell that had been deleted could get undeleted. Can you have a cell that gets deleted get undeleted? No, we have no cell reincarnation here. And so deletion isn't really breakable. You can't really mess up, or at least from what we know about deletion, you're not going to get the cell back. Nothing is going to sort of make this one get broken. Once the cell's dead, the cell's dead. And that is why I put the second one here. Because the second one has a similarity in terms of the breaking it part. So the second one is based on a fact that you certainly know, which is that B cells require T cell help in order for those B cells to proliferate, to class switch, to make high levels of antibody, differentiate to plasma cells, all that good stuff. So remember that B cells largely require T cell help that we see here. The second type of peripheral tolerance mechanism that we know about is called clonal helplessness. And with clonal helplessness, we might have a B cell that is self-reactive. However, the T cell that would have helped that B cell got deleted in the thymus. Because in the thymus, all the self-antigens are present. We have a whole lot of T cells. And so some of our B cells are never going to be able to get help because their cognate T cell got deleted in the thymus. And so that B cell might interact with antigen in the periphery, but because it doesn't get T cell help, it's not going to be fully activated and may in fact be energized. Given that other cell types besides B cells do often require T cell help, you can imagine this as relating to things other than B cells, although usually people talk about B cells for this one. And so the idea here is that because T cell central tolerance is so good at deleting against so many antigens, that ends up spilling over because you're getting rid of the B cell help. Yep?
Why do all these start with clones?
Because that's how I learned them. Which is how I learned them. So, can we break helplessness?
No.
Yeah, you talked about it, and that's what air.
Okay, but we're now doing a peripheral tolerance. Again, if you don't have air, like you got all sorts of problems. Yeah.
Okay, I would say no, because if that T cell doesn't exist, why would it randomly exist?
Yeah, we're not reincarnating T cells either. So again, we can't make that T cell re-exist just like we can't make the deleted cell exist. And so again, this one doesn't break in this quite the same ways that some of the other ones are going to break. Yep.
How is this not just an extension of central tolerance?
I mean, yeah, it's because the B cell has made it into the periphery, and so its lack of responsiveness is in the periphery. But yes, as I hope you have seen throughout this entire semester, none of our great strict lines between some of these things in the immune system are all that black and white. Just like we saw last time with innate versus adaptive. So, yeah, sure. It's messy. All right. So there's two, and you can see how you kind of already knew them both. The third one I'm going to tell you about, you also basically already know about. It is known as clonal suppression. And clonal suppression is a fancy way of saying Tregs do their thing. And so we can have situations where we have self-reactive cells that could be self-reactive in a peripheral tissue. However, regulatory T cells can inhibit those other cells and thus suppress that autoreactive response. And so if we have a lot of regulatory T cells, then we're going to suppress this response. In the literature, there are other subsets of regulatory cells that have been described, like B-regs and things like that. I am not going there, but just so you know, there are other ones that have been described, and they would also fit into clonal suppression. It turns out that you kind of know about clonal suppression already. I'm going to tell you a little bit about clonal suppression. But it's going to segue us into the fourth mechanism. And the clonal suppression and the fourth mechanism also kind of bleed into each other in a bunch of ways. The way you break them really bleeds into each other. So I'm not even going to deal with breaking this until I get to the other one. Then we can do those together because it's the same thing. But how many of you guys have heard of things to be done for kids with super severe peanut allergy? What do you do for a kid who has a super, super, super severe peanut allergy? Yeah.
Teeny tiny amount of it at a time, and then over like a long period of time, somewhere around 6 months in the minimum, I think you increase the dose until they're having somewhere, I think, 1 peanut a day, and then from there it's easier to kind of go to like 2 and 4, and then you stop at around 8.
So there's this thing where you sort of are sort of delivering the antigen at a very small level. Do you know how they deliver it?
It depends, I think, on the patient. Like the way it happened with my little cousin is it was orally.
So there's this process known as oral tolerance. It turns out that if you, so first, let's just see, we've got this mouse. And the mouse is injected with an antigen called ovalbumin. And it's injected sort of just in, the intramuscular in the skin. Yeah, they don't really tell you where. You just give the mouse ovalbumin. And then you look later, does it make an immune response to ovalbumin? And the answer is yes, it makes a big immune response to ovalbumin. It turns out if you have previously fed that mouse ovalbumin, and then you do this injection, the mouse no longer makes that response to ovalbumin. It makes a much reduced response. And so it seems as though when your first exposure to an antigen is through the oral route, you end up making more of a Treg response. In fact, Tregs were first described in the GI tract and were only then described later. And in some ways, that kind of makes sense. An antigen that you first encounter through the GI tract is probably a some kind of food, and you probably don't want to make a big response against it. And so there is this active downregulation of inflammatory responses to food that you first encounter in the environment, and actually a turning on of regulatory T cells in that environment. Obviously, I know that then people want to ask lots of questions about food allergies and things, because obviously there are lots of questions this raises. The answer is generally we don't know. I will also warn you for the rest of the semester with the clinical stuff. A, I am not an MD. B, I am not your MD, and I do not know your personal health history. So at some point, if you're like, well, why did my doctor give me this medicine? I'll be like, that's a great question. Why don't you ask them? So just so you know, that there's some point where that comes up every time. So there is this idea that oral tolerance can be helpful, and in fact, oral tolerance therapies for kids who have the super severe peanut allergies do work pretty well. But we're talking here about like a kid who can't be in a room with where somebody opens a bag of peanut M&Ms, or else they're going to go into anaphylaxis. Like there are peanut allergies that are that severe, which can be really a problem if you go to kindergarten or something. And so oral tolerance can make it, that kid's not going to be eating peanut butter sandwiches every day, but that kid can survive in an environment where there might be some peanut allergen. Yep.
Most of the time, isn't this temporary though? Like, don't you have to consistently ingest the antigen in order for this to be successful?
The tolerance?
Yeah.
Not always. Sometimes it can be persistent. There is actually a hypothesis. There's not great data to support this yet, but it does I'll explain it to you, and it sort of like makes sense. That one of the reasons for the rise in peanut allergies is because a lot of baby lotions now have peanut oil in them. And so more and more kids are first encountering peanut allergen through the skin instead of orally. And as a result, they're making this control response instead of making their first peanut response being an eating a peanut response. And so there might be this question of if we either feed kids something with peanuts earlier and or get rid of the peanut antigens in their lotions, will that make a difference? So again, a practical way that this comes together. So this is related to the fourth mechanism of tolerance. And the fourth mechanism of tolerance has to do with control of CD4 helper subsets. Given that Tregs are one of the helper T cell subsets, you can see why this is kind of related to the other one. So officially, this one more is thinking about like a balance between Th1 and Th2, but again, you can see how it gets messy because Tregs technically could be a part of this. And this one is known as clonal deviation. And so the idea with clonal deviation is that for every particular type of response, to some antigen, in this case it's the helminth antigen, one type of T cell will give you a protective effect, whereas another type of T cell will give you a damaging effect. And so the idea with clonal deviation is that T cells are pushed towards protective or non-damaging responses instead of being pushed towards damaging responses based on the types of cytokines that are produced. As you might imagine, the way that we can think of a way this gets messed up. So let's imagine you encountered some antigen. And you encountered that antigen, and ideally you want to make a Th2 response, because that's the good protective kind. Is there a circumstance where you could make a Th1 response instead by mistake? So I see lots of head shake, yes. Why? How? Yeah.
If you have the MHC class that binds to, like an MHC class 2 that binds to the thing that induces the CD4, or induces the Th1 response instead of Th2 response.
So MHC is not as much the issue here. Not so much about MHCs. Yeah, Dallas. Yeah, so what happens if your IL-4 response happens to be inhibited on the day that you encounter that antigen? What happens if you just happen to be making a ton of interferon gamma for some other reason that day? Maybe you're also fighting a viral infection that day, so there's a lot of interferon gamma around. So the cells get confused and start making Th1 because they get interferon gamma from the NK cells that are dealing with the viral infection. They pick up the wrong cytokine, they get pushed the wrong direction. They are deviated incorrectly. And so we can imagine, clonal deviation. And so what we can imagine is that we can break either deviation or suppression by providing inappropriate cytokines when we have the response being made. And so what you can sort of imagine here is that this is a case of really bad luck. You happen to be making one kind of response, another antigen comes around, you end up making the wrong CD4 subset response and getting a pathogenic response where you would have had a protective response. And so this could be a way that you could break deviation. This is also a way that you could break suppression. So this is one where they really come together. Students have often in the past found this one a little bit complicated. There has been a group of super amazingly cool papers that actually show a disease state happening because of this. It's really cool. So the next few slides, I'm going to just sort of tell you a little bit about the model from those papers. The goal is that it helps you understand this better. Let's hope I don't fail and make you understand it worse. But we're going to try. And so this is one situation where there are some mice that have either are wild type or that have a mutation. In wild type mice, they can be infected with a couple different types of viruses and they are fine. If, however, and in fact, this mutant mouse, based on its genetics, if it's infected with one version of the virus, it's all good. But if this mutation, which changes the cytokines the mouse makes in response to the virus, so we've got changed cytokine because of this mutation and a specific strain of the virus that this mouse is infected with, and this mouse ends up with Crohn's disease. And so the mouse was unlucky enough to get orange virus instead of blue virus, and was unlucky enough to have this mutation. And that sort of combination of things led to changes in cells in the GI tract, changes in gene expression, and problematic responses in the intestine. And so this is one pretty famous example where basically this mouse is having its response to the orange virus be deviated due to its genetics. The other story that's probably the more famous of these stories has to do with this model of celiac disease. And so with celiac disease is actually like tricky for so many reasons in immunology. In fact, it's even controversial whether we call it an autoimmune disease because the antigen is something that comes in from your diet but that then is modified. So it's in some ways like an allergy because it's an outside allergen, but you had to modify it. So like, is it self or is it not self? Anyway, that's a story for another day. We're doing allergies later. But so there is this antigen. Typically, the antigen is picked up by some kind of dendritic cell. That dendritic cell presents to T cells, and that dendritic cell generally makes cytokines so that we get Tregs. And so usually people have this antigen and they make Tregs that respond to it. However, sometimes people happen to have a viral infection at the same time as their first ever exposure to this antigen. So again, super bad luck that you happen to have a viral infection, this particular reovirus, that day. As a result, you start to make T cells that are Th1 cells against the virus because you need to get rid of the virus. However, the cytokines that are being made in this process spill over to the dendritic cell and eventually lead you to make Th1 cells against the dietary antigen, instead of making the Tregs that you should have made. And as a result, you now have an active immune response to this dietary antigen, this gluten, that you would not have had before. And so again, this is like super unlucky that you actually need to have the right MHC to even present this peptide. And you have to have gotten, this type of viral infection like the same time as your first ever exposure to the antigen. So it's a whole lot of bad luck. And I always kind of hate doing these lectures because sometimes people have different conditions in the class, and basically I'm just sitting here being like, wow, look how unlucky you were. And so I'm feeling terrible as I'm talking through these slides. Let's see, Lexi.
Is that the only way that you can get celiac? Is it that condition, or like that,
There's so much that can be said. This one is really well documented and well described.
is it the most common?
This one is really well documented and really well described. That's kind of what we got at this point. And so this paper, you can see, is 2017. They have a follow-up that came out this year. So it's from the guy who does a lot of it is Terrence Dermody. He's at University of Pittsburgh, and he's done a lot of this model. And so there's a lot that we don't know. It's probably not the only way. Frequency we don't know, but again, this is one model where people have really worked it out very nicely. Yep, Michaela.
So how is it that you can develop celiac?
Because it's sort of a question of, do you ever get, so if you were to develop later in life, really what you're seeing is somehow these cells are getting deviated. They're going from Tregs to Th1s, or you're starting to make a Th1 response off of the naive cells. So there are some cells that are getting suddenly the wrong cytokines and starting to make that response. Yep.
So is this something that's like unique to celiac disease, or can it apply to like allergies and food?
It could potentially apply to many different types of conditions. Again, this is a very well-described model where people have really been able to say it's this virus and it's this, and really worked it out. But conceptually, we think this is how a lot of similar processes work. Yep.
Please don't hate me, but does that mean you can like get celiac randomly at any time?
Potentially. So, okay, so there's sort of two models in terms of allergy and autoimmunity. We're not even in the allergy day. And whenever we get to the allergy, there's 2 days of allergy, and somehow I end up having to diagnose everyone's problems whenever we do allergies. So we're not even there yet. We're coming up with the scenarios. So they're kind of two general patterns. So imagine that you have a cell that is responsive to your favorite allergy antigen. Whatever, or your unfavorite allergy, whatever antigen, okay? So let's imagine that cell should become a Treg. And let's imagine that cell gets an itty-bitty amount of IL-12. Maybe you're going to make a couple Th1 cells, but not enough where you're going to feel bad, right? Not enough to really do something. Now imagine next month you get a little bit more IL-12, and a little bit more, and a little bit more, and a little bit more, and a little bit more. And so you can see sort of this progressive autoimmunity or this progressive allergy where the cells are getting further pushed and further polarized, which would probably be the adult development of allergies and autoimmune diseases. Typically in those adult developing diseases, you tend to see a somewhat worse outcome just because the cells have kind of been pushed over the edge multiple times. And so, for example, most of my atopic conditions have been adult developing, and so I'm just like, stop it, mast cells. That's okay. You could also imagine that the cell starts out making an inappropriate response and then gets sort of pulled back by suppressive cytokines. That tends to be what happens with sort of the ones that kids outgrow. And so it could sort of go either way based on what we're seeing with this deviation. And so here's the model that they came up with that paper. The gluten will come in, and you're getting this infection with this reovirus at the same time. The dendritic cells are thus making inflammatory cytokines to push T cells to Th1 cells, and that eventually leads to the destruction of the excuse me, the intestinal wall. And so this is sort of one of the ideas that we have. All right, so do we get deviation? I would say that that is like the most engaged I've ever had a class about deviation, so I'm thinking maybe you understood. All right, so those are 4. The 5th one's a pretty big one. The 5th one is one that people think about a lot. And again, it's one that you've already learned about. The place where it gets a little tricky is in how you break it. So, Mark, you already mentioned this. How might we do peripheral tolerance? It's anergy. And specifically here, we're thinking about T cell anergy. And so this one is known as clonal anergy. And so you already know some things about clonal anergy. You know that our T cell requires signal 1 and signal 2 in order to be activated and to proliferate. If that T cell receives signal 1 alone, which might happen if it's seeing a self-antigen, because there won't be production of our co-stimulator, because there's no pathogen-associated molecular pattern, no PAMP or MAMP, to make our antigen-presenting cell make this co-stimulator. So this is all the T cell can get, is just that self-antigen that's being presented. The T cell is going to become anergic, and even if that T cell gets signaled later on, it remains anergic. And so we've seen this process before. The problem with anergy is that there are a few different ways that anergy can be broken. One of those ways is shown here. So we can imagine some kind of antigen-presenting cell, like this resting tissue dendritic cell, presenting self-antigens. Remember that our antigen-presenting cells, particularly those that present on class 1, are presenting self-antigen at all times. So it's just going to be presenting some antigen like this. It doesn't make any signal 2, any B7, because it doesn't have any pathogen. And we can see it interacting with the T cell and energizing that T cell. Notice here that our dendritic cell is not presenting one copy of MHC with peptide. It has a bunch of MHCs on its surface, all containing peptides. Now, if we have an infection that happens, if this, we have a microbe that infects this DC, you can see that the DC was presenting self-antigen. It's going to start presenting microbial antigens, which is this brownish thing that's being presented on MHC. It's going to start making B7 because there's a PRR stimulus in that microbe to start having this made. But it's unlikely that every single MHC molecule on the whole surface of that cell is going to have microbe antigen or pathogen antigen. It's entirely possible that some MHCs on the surface of that cell will still be presenting self-antigen. The B7 molecule doesn't know which TCR it's supposed to hang out with. It, in fact, is diffusing randomly throughout the cell membrane and is going to bind to whatever ligand is close by. And so you can see that this T cell that is specific for our microbial peptide is getting MHC plus peptide and also B7 and signal 2. And we want that. We want to turn this on, and we want to get rid of this terrible microbe. But unfortunately, we also can see that that B7 also could interact with CD28 on this self-reactive T cell that's reacting with this self-antigen. And we could have just turned on our self-reactive T cell. And so this cell in a perfect world would have been energized, but because, again, of sort of bad luck here, this cell got turned on, and we now have a self-reactive response and autoimmunity. Yep.
So why isn't this one temporary? Like, why isn't it that just when the pathogen
because when we talked about immune response kinetics last week, and one of the things I told you is that naive cells and activated cells are different. And one of the big ways that naive cells and activated cells are different is that naive cells require signal 1 and signal 2 for activation. Once a cell's been activated, it doesn't require signal 2 anymore. So once it gets this first signal 2, it never needs it again. And it can now go out into peripheral tissues and start destroying things because activated cells no longer need signal 2. Yep. Good question. Good question. Because we have all of these things happening at the same time. So we've got all 6 of these mechanisms happening at the same time. I know there's so many cells, and there's so many antigens, and there's so many responses. I know. But yeah, really the answer is because we've got all of these sort of overlapping each other. Yeah, Nick?
Wouldn't this happen pretty frequently because cells constantly
Potentially. I mean, part of it has to do with sort of how much of the microbe proteins are being made and what proportion of the MHCs are filled with microbe versus self peptide. And sort of what proportion of them are going to be able to pair with a B7. So you can think about it, if say 1% has self, maybe all of that, if 1% of the MHCs have self and 99% have microbe, then maybe that 1% isn't going to pair with a B7. And so it's kind of a math problem. Yep.
So we talked about immunoproteasomes earlier in this semester.
Mm-hmm.
I know you said that those make it easier to like present certain epitopes. So does it also stop like cell peptides from being presented as to a certain extent?
You should do a PubMed search for someone named Udell, Yewdell, JW, Jonathan W. Udell, because he has a model that says that it does, but mostly that model is coming from Udell, and there isn't a lot of other people talking about that. And so I have seen a model that tries to make that claim, but it's not always described. And so if you look at something called the DRIP theory by Jonathan Udell, it will address that. So you guys are all pretty sure you're going to die of broken anergy right now. And guess what? This is only one way anergy gets messed up. I am not your physician, nor am I anyone's physician. Maybe I'm a mouse's physician. That's all I got. So the other major mechanism by which anergy can be broken is something called molecular mimicry. And so remember that when MHC is presenting the peptide, some portion of the peptide is sort of pointing down into the peptide binding groove of the MHC pocket, and some portion is pointing up. You could imagine 2 different MHCs would have pockets that were slightly different, and so they might bind to different portions of the peptide, but say the portion of the peptide that is sitting up, going towards the T cell receptor, might look the same. And so you can see this pathogen peptide plus MHC and this self-mimic peptide plus MHC look the same as far as the T cell is concerned. The T cell can't see in this cross-section and see that this part's different. And so when we might see a situation where the T cell is activated by a pathogen peptide that has been, say, presented after phagocytosis by this macrophage. So we're presenting, we're making B7 because this is foreign. We're turning on that naive T cell. Well, now our T cell, it doesn't need no signal 2 anymore. It's an active cell. And so if it were to go into the periphery and interact with some type of self-peptide that may have a similar response, even though this self-peptide is not going to be presented along with a signal 2, the T cell does not need signal 2 anymore. It's already been activated with signal 2. And so that T cell can be turned on. And that T cell can act against those self cells. And so we can sort of imagine it looking something like this with cross-reactivity. So we could imagine that in childhood we have a viral infection where these happy purple T cells kill the virus. And so we get over that one. It was random runny nose number 7 you had when you were 4. But it could just so happen that those T cells happen to react with MHC plus peptide that is presented on beta cells of the pancreas, particularly the insulin protein, which is actually pretty frequently targeted in beta cells. And so those beta cells of the pancreas, which are these happy red cells, they get dead because those CD8 T cells kill them, and now the patient has type 1 diabetes. And so type 1 diabetes is, in fact, a completely autoimmune CD8 T cells killing the beta cells in the pancreas. Type 2 works just slightly differently. But this is exactly what happens in a type 1 diabetes situation. And the problem is that this antigen that is on those pancreatic beta cells cross-reacts with some other antigen that was present at the beginning. This can happen. Here, let me get to the next 2 slides and I'll answer. This can happen. So here you can see in blue MHC and in green a peptide. The red and the yellow are actually the T cell receptor. And so you can see this is actually sort of the normal binding for the T cell receptor. There are situations where T cells will end up binding a little bit off. So here you can see sort of this is a slid over T cell receptor binding to MHC plus peptide. Or here you can see that it's binding at a totally wrong angle to MHC plus peptide. And sometimes that can lead to cross-reactivity. These are actually 2 different T cell and MHC peptide things that are actually found in multiple sclerosis patients, where we see CD8 T cells destroying myelin and reacting against myelin. And so you can see that in some cases we can have sort of inappropriate angled responses. And further, just so you know, molecular mimicry is not limited to being a T cell problem. There are plenty of situations where there is a B cell molecular mimicry as well. So we can see, for example, situations where you might have antibodies against a particular antigen. A really famous one is this antibody against the streptococcal cell wall carbohydrate. And it turns out that one particular antibody you can make against the streptococcal cell wall is cross-reactive with heart tissue, causing a disease called rheumatic fever, where antibodies actually lead to destruction of heart tissue. And so, again, same process of molecular mimicry, not just a T cell problem. Let's see. Mark, what was your question?
So there are like 25 trillion possible anchorosms, or like more than that, right? Or like peptides, right? So how is it that this occurs more than like once? Like how is this such a common thing that happens? First question.
Because while you can think about all of the possible proteins and peptides that exist, if you then sort of subset it based on what actually makes sense biologically and what actually works functionally, there's a lot more overlap.
And then second question is, like specifically for diabetes type 1, do they know like the specific virus?
No. No, they do not.
How do they know it's a virus then that causes it? Like how do they know it's specifically an upper respiratory virus, but not know which virus it is?
I'll get there. I can come to that. There's much to be said about that. Yeah.
Is mimicry more common in T cells compared to B cells because like the specificity of the transformation?
I don't know that we can fully measure that. I can see how it would make sense that we would think that, but I don't know that we could. I don't know if there's a great number to measure that. So the moral of this story is that what you might notice is that there, in many cases, does seem to be some type of role of infection in autoimmunity, where having some infection may break some form of tolerance, especially in the anergy situations. And so in some ways, what you can think about with the anergy situations is that it's a race for a lot of these T cells. Do they get energized by seeing self with no signal 2, and so they get energized and they're totally off, and we're done? Or did they happen to get turned on before they ever got energized? So it's sort of this race of, are they going to get energized or are they going to get turned on first? And so if they happen to be unlucky, remember this probably has something to do with, actually not probably, this certainly has something to do with your specific MHC type and which antigens you are susceptible to having problems with, and sort of a lot of luck issues. Which infections do you get? What order do you get them in? All of those types of things. There are a lot of associations with infection and autoimmunity. So some of the problems, so this sort of gets at Mark's question about, why don't we know exactly which virus it is? And usually you don't sort of sneeze and the next day you have diabetes. Oftentimes there's like maybe 10 years or more in between the two acts. And so if random runny nose number 4 when you were a kid was never diagnosed, how the heck are you going to know what virus you had 10 years ago? And so you can't go back in time and run the immune system back.
How do they know it's upper respiratory?
They didn't really. So what they do know is they can say most patients with diabetes have evidence of infection with either Coxsackie A, Coxsackie B, echovirus, or rubella. All of those happen to be upper respiratory. So they can say there is statistical association with having been infected with this, having this HLA, and getting diabetes. But what the initial triggering event was, we cannot figure out. We cannot go back in time and play things back. For autoimmunity, and in fact for allergy as well, we know a lot about the process that keeps the allergy going or keeps the autoimmunity going. But the initial hit that starts the whole process of going wrong, we don't usually know because we can't go back in time and re-see what happened. And so Jill can tell you, in emerging infectious disease, I spent a lot of time talking about how we don't always track every type of infection, and we don't necessarily know about everyone's infection history and all the details, and this becomes a problem. And so that's one reason why that reovirus celiac story that I told you is such a big deal, because we actually figured it out in that case. But in theory, it could be another GI virus that also messes up the cytokines in that same location. We cannot say that there isn't another virus that messes up cytokines. And maybe in some patients it's Coxsackie A, and some patients it's echovirus. And they both lead to cytokines messing up the T cells. So there are a lot of things that could be going on here. Yeah?
But if they know that there's something that's mimicking self-peptide, why aren't they able to just figure out, okay, what's the peptide sequence that's mimicking the self-peptide and then match that to a virus?
Because humans are outbred and we all have so many different MHC molecules. And so you could only do that if every patient you saw had the exact same MHC molecule and the exact same pathogen. Then you could start back calculating that. But once you actually realize that every patient has 6 possible MHCs and it just gets...
How are they able to associate just 3 MHC classes, or 3 MHC?
Those are the most predominant ones that are seen in patients. So they are the ones where if there are certain situations for all sorts of different autoimmune diseases where there are some that are sort of statistically associated. It's not 100%. There are people who don't have DQ2 or DQ8 or DR4 who still get type 1 diabetes. These are just the most frequently found. So it's an association, but it's not a perfect thing. And so what you can see, however, what I hope you see is that there might be a role for infection, but we can't always trace back what that infection is. And this is very much something people are working on, and that's why that celiac paper is so cool. So at this point, you've seen 5 of the 6 mechanisms of peripheral tolerance. I'm going to start introducing number 6, and then we're going to leave off to next time when we are going to learn all the reasons why the 6th one is always everyone's favorite, and it's very, very cool. Okay? So the 6th one involves some anatomic locations. These are the most famous of those anatomic locations. This one is called either Clonal Ignorance, because we want to make it sound similar to the other ones and have clonal in the name, or often it's called immune privilege. And so the anatomic locations that are really important here are known as the immune privileged sites. And so these are the most famous of the immune privileged sites. So I want us to sort of leave off here thinking about these immune privileged sites. So if you look at this list of organs, if you look at this list of anatomic locations, what do all of these things have in common? Yeah. They're all super important evolutionarily. So what do you mean by that? Yeah, if one of those gets messed up, you're kind of done. Right? And so it turns out that all of these locations specifically have a lack of full immune responses. Because if there was too much immune response, if there was too much inflammation in these locations, we would see a detrimental effect that would be bad evolutionarily. So we have evolved. If we started to have so much inflammation that fetuses couldn't live, we would not survive as a species very well. Similarly, we could imagine having so much inflammation that the brain gets damaged, so much inflammation that the eyes get damaged, that this could be very detrimental. And so all of these anatomic locations have evolved some type of reduced immune defense, which we will talk more about next time, to sort of get rid of that process or get to lessen their chance of being damaged. And they're known as the immune privileged sites. One thing that I will say is that there's sort of the classic definition of immune privilege, and then we're learning more about it and realizing it doesn't quite work the way we used to think it worked. And so that's a big part of what we're going to talk about. And it also certainly has some big cons, and we're going to talk about those as well. And so we will start off with more details about immune privileged sites on Wednesday.